I would now like to invite Mariam, our next speaker. Mariam Mohammad Hashim is a research associate at Lahore Medical Research Center. Her primary focus and expertise lie in conducting research using in vitro and in vivo modeling techniques. The theme of her presentation is the associations between HLA allele frequencies and the severity of COVID-19. Mariam, it's up to you.
Good morning, everyone. I hope my screen is visible now.
Yeah.
All right. First, before I start, I would like to thank Society for the Study of Evolution for giving me the opportunity to join as a speaker. So I am Mariam Hashim from Lahore, Pakistan, and my primary expertise lie in conducting research using in vitro and vivo models. And the title of my today's talk is Associations between HLA allele frequencies and the development and/or severity of COVID-19. But before I start, I just want to let you guys know that this data was collected at the beginning of COVID-19 pandemic. And by that time, there were very few studies which were reported. So we will just start with the basic introduction. As we all know that human leukocyte antigens, they are important part of the immune system, and they are usually controlled by the group of genes which are located on chromosome 6. And they are involved in encoding cell surface molecules, which are specialized to present antigenic peptide to T cell receptor on T cells. And if we talk about MHC, which is major histocompatibility complex, they present antigens and they are divided into two main classes, which are MHC class 1 MHC molecules and class 2 MHC molecules. If we talk a little about class 1 MHC molecules, they are presented as transmembrane glycoproteins on the surface of all nucleated cells, and they are intact. The intact class 1 molecules, they consist of a heavy alpha chain, which is bound to beta 2 microglobulin molecule, and the heavy chain of class 1 molecule is encoded by multiple genes which include HCA, HLA-A, HLA-B, and HLA-C loci. And if we talk further about class 2 MHC molecules, they are usually presented on professional antigen presenting cells, and they consist of two polypeptides, alpha and beta chains. And both polypeptide chains are encoded by genes such as HLA-DP, DQ, or DR region on chromosome 6. So since HLA genes, they are highly polymorphic, meaning they exist in many different variations or alleles within the human population. The diversity of HLA alleles contributes to the overall diversity and effectiveness of the immune response, allowing the immune system to recognize and combat a wide range of pathogens. So at the beginning of COVID-19 pandemic, many worldwide researchers have been investigating various factors that were mainly influencing the development and severity of the disease. And many genetic factors were concluded, including variations in HLA alleles. They were found to play a role in how individuals respond to viral infections like SARS-CoV-2, which was the main virus responsible for COVID-19. So it is worthy to note here that HLA genes, they play a significant role in the immune system, and they are not the sole determinants of disease outcome. However, COVID-19 is a complex interplay of various genetic, environmental, and lifestyle factors, and ongoing research is necessary to comprehensively understand the disease mechanisms and improve clinical outcomes. The COVID-19 outbreak in China was deemed a public health emergency of international concern by the World Health Organization on January 30th, 2020, and the danger was higher for nations with the weak, healthy immune system. So the objective of my study was to see the relationship between HLA alleles and the severity of COVID-19. So since we all know that HLA, they play a huge role in our immunity, the major differences in alleles could promote the development and or severity of COVID-19. So if we talk about the already published literature, it was found that individuals with the genotype of HLA-B4601 genotype, they are more at the risk of developing COVID-19. So moving towards the materials and methods, population density in different countries and the statistics of COVID-19 cases were obtained from a database of World Health Organization. And the purpose was to assess the densities of population and the incidence to observe the genetic association of the disease. And meanwhile, the studies which were reporting the frequency of HLA alleles in COVID-19 patients were included and the rest were excluded. And since I've started this, since I have already mentioned at the start of my presentation that by that time, very few studies were reported. So the only studies I can get was from China, Taiwan, Hong Kong, Vietnam, and United Kingdom. And later on, these frequencies were compared with the frequencies of these alleles in those general populations as mentioned in the databases of compare global HLA allele frequencies. The link is here. The link is given here, and allele frequency net database. And statistical analysis was done using SPSS. So if we talk about the results majorly, if you guys can see that the bar graph represents the population and the population density and the percentage of cases. So this figure shows that India and China, they are the highest high density populations, and the percentage of COVID-19 patients in India was very low by that time as compared to China. And USA population density was low, but the percentages of COVID-19 patients were very high. And in Japan, Japan was also considered to have the highest cases of COVID-19. And if we talk about the population density and percentages of deaths, the population density and percentages of deaths was varying from population to population. And India and China were high density populations, but the death rate in China was comparatively high as compared to India. So these are the common HLA alleles in Taiwan, China, Hong Kong, Vietnam, and United Kingdom. So if we see this allele, particularly this allele, HLA DRB10701, this was the allele which was found most frequently in China, Hong Kong, and UK. However, this allele was not prevalent in countries like Vietnam and Taiwan. And if we talk about the second allele, HLA-AB 15 and HLA-DB DQB10601, this blue graph represents HLA-B allele, which was found to be frequent in China, Hong Kong, and Vietnam, whereas this red bar graph is depicting the another allele, DB DQB1, which was also prevalent in China, Hong Kong, and Vietnam. And if we talk about further alleles, which include HLA-B1301, DRB11202, DQA101, and DPB10301, they were prevalent they were less frequently found in Taiwan, but the China population in China showed the highest ratio, and Hong Kong was the highest in terms of this allele, which is DQA101. So these are the allelic frequencies, particularly particularly we can see in Taiwan. I hope it's visible. It's blurry, but I think I can explain this a bit better. So if we see, these are the alleles, A2402. This allele was the most frequently found allele which was present in the healthy population. But according to the databases of WHO and the allelic frequency database, this particular allele, A30 highlighted in red, was found to be linked with the severity of COVID-19. Moving further, if we talk about HLA-B, the allele which was found healthy in overall population was this one, but the ones highlighted in red were found to have been linked by the COVID-19. Similarly, if we talk about HLA-C, this particular allele was the highest in general population, but HLA-C 1502 was found to be linked with the development of COVID-19. In case
Mariam, sorry to disturb you. Your voice is very low. It's difficult to hear.
I hope it's fine now.
Yeah, now it's okay. Thank you.
Okay, if we talk about the MHC class 2 alleles, no alleles were found to be linked with the development of COVID-19. If we talk about DQA1, this particular allele, which is highlighted in red, was linked to was found to be linked with the development of COVID-19. Similarly, DPB1 frequency, the overall graphs which are showing the ones which are highlighted in red, they shows that they have been linked with the severity of COVID-19. If we talk about particularly allelic frequency in China, and these are the alleles which are present in general healthy population, and they were in rank of 18th to 20th frequency group. And this particular alleles in A31001 and A301 was found to be the most prevalent in China, and with the individuals in class of HLA-B alleles, B1301, B15002, B13002, B46, and B5401 was found to be linked with the COVID-19, particularly in China. Likewise, in case of HLA-C, C0801 was found to be prevalent with the COVID-19. And in case of DQA1 was found to be associated with the development of COVID-19. So these are the allelic frequencies, alleles which were mainly reported that time, causing the development or severity of COVID-19. So if we take a few cases from Hong Kong, these were the major alleles, namely B1502 and B1301, which were linked with COVID-19. If we talk about HLA-C, C801 and C1502 was found to be linked with the development of COVID-19. In case of DQA1, this particular allele, which is 101, was found to be associated with COVID-19. DQB1, DP1, and DRB1. These all highlighted in red were found to be associated with the development of COVID-19. Likewise, if we talk about Vietnam, these highlighted in red, B1502 and B4601, these were the alleles which were associated with the high risk of COVID-19. The rest of the alleles were found in general healthy populations, and none of them were found to be associated with the development of COVID-19. If we talk about the MHC class 2 alleles, which is DRB1, DRB1 1202, 0301, and 0301 was found to be associated with COVID-19. And for DBQ1, 50201 was found to be the cause of COVID-19 in the overall general healthy population. So this is the allelic frequency in UK. The major cases were reported in UK, but we could not find major alleles associated with the development of COVID-19. And that may be because of few cases were reported that time. But I'm still working on the extension of this project, and I'm working on developing heat maps for this particular data. But they're still in process, and the data is still not published. But these alleles, HLA, in case of HLA A0301 and DRBV1, which are highlighted in red, were found to be associated with COVID-19. So the growing recognition of genes and existing evidence of those responsible for host immune response by involving the MHA HLA. This article helped to explore the relationship of genetic makeup of individuals with disease susceptibility and to some extent disease severity. Multiple HLA alleles were identified to be associated with the risk of development of COVID-19. However, further research is still needed, and in silico studies, they are still needed to conclude the exact cause, and individuals that are at high risk could be recognized by understanding possible variations of HLA in relation to progress of COVID-19 progress. In addition to testing for COVID-19, HLA typing could be utilized in routine to avoid potential worsening of disease, and patients could even prioritized for eventual vaccination. So this was a short study about COVID-19 in relationship to HLA alleles. If you guys have any question, you may ask.
Thank you, Mariam. Thank you so much. You had perfectly timed it to 12, even though you could go up to 15. Thank you. We already have questions. Sorry, no, this is not yet. Sorry. Okay, so here's a question from Sutirth. He says, different populations are showing different alleles at higher frequencies. Do you have any hypothesis as to the potential mechanism behind this variation?
It could be, according to me, it could be the individuals which are having a weak immune system. They might be at the higher risk of developing COVID-19. And that all depends on the lifestyle factors, the genetic makeup, I guess.
Okay. Any other questions? No? Okay. in which case, I would like to thank Mariam. Thank you so much for an interesting talk. And this has been a very topical talk in a sense, given what we all have gone through over the last couple of years. So thank you so much.